So today we're very pleased to welcome Dr. Irina Buckle. She's coming to us from Mater Research in Australia, and she's an immunologist in the Professor Josephine Forbes Laboratory at Mater Research Institute in Brisbane, Australia. She focuses on the development of disease-modifying therapies for T1D, specifically focusing on the receptor for advanced glycation end products, or otherwise known as RAGE. During her PhD under the mentorship of Professor Hamilton Williams, Professor Regina Thomas, and Professor Raymond Steptoe, Dr. Buckle worked towards development of antigen-specific immunotherapy for T1D in preclinical models, and this approach is currently being translated for human use. The title of her talk today is RAGE Targeting Therapeutics for Type 1 Diabetes. Welcome, Irina. Thanks so much for joining us. We've been following this story, as I was saying off camera, since our first interview with Josephine Forbes just before the pandemic. And it's just a fascinating tale that and story that your group has continued to, develop. And I can't wait to hear more about what's going on right now.
Excellent. Thank you very much for the kind introduction, Monica, and thank you for the invitation. Very pleased to talk to you today. And as I mentioned to you, IDS, it's a great initiative, and you guys do a great work bringing us all together and letting us discuss what we all think our research is fantastic, but everyone's research is
I
really
will agree.
fantastic.
It is. I mean, there's so much talent in this field, and so many different incredible scientists at many stages of their career across the globe examining this complex disease through different lenses. So it's our pleasure to have you. Thanks.
Thank you. And we really do have a wonderful community. Type 1 diabetes, I think, is quite unique in its sense. But so today I'm going to touch on some of the work that we are currently doing and some of the work in the development in our laboratory here in Brisbane in Australia. And we're all about RAGE and RAGE targeting therapeutics for type 1 diabetes. So our audience is very well versed in what type 1 diabetes is. So I do not have an introductory slide, but I'm going to jump straight in because we think that receptor for advanced glycation end products is very relevant in a number of inflammatory conditions, and including type 1 diabetes, of course. So it is an immunoglobulin type receptor that downstream signaling upon the ligand binding, and the ligands, there is a number of, my slide represents just a couple, but there's a number of ligands that bind to that receptor, leading to the downstream signaling and the activation of kappa B and induction of generation of reactive oxygen species that of course leads to inflammation. It is quite important in this scenario, particularly in type 1 diabetes, that some of those ligands are released in from the inflamed pancreas, injured pancreas, and of course dendritic cells also release some of those ligands. And there is some evidence that there is a DC maturation and the T cell proliferation, RAGE upregulation. It's all immunological profile on immune cells of RAGE is we're still trying to decipher and know a little bit more about, but the truncated isoform soluble RAGE, and of course Sherman's work, and you've already heard from Josephine and Sherman on that, and I'm going to touch on as well. So there is a number of therapeutic strategies that we are exploring, and that of course ligand receptor binding is one of them, and blocking that downstream signaling in order to prevent some of the inflammation that is caused as a downstream. We know there is some evidence that RAGE is upregulated in at-risk islet autoantibody positive individuals, and it is associated with progression to type 1 diabetes. And Professor Harold in the past decade some of his research has touched on that a little bit. And of course Josephine's work with Finnish groups talking about RAGE SNPs that are predictive of type 1 diabetes, showing further evidence that it is very important receptor I'm not sure why my slides aren't progressing. Oh, sorry. Receptor in the progression of the disease. This publication that Sherman is, and he can pipe up if I'm wrong, but I believe it's currently in the review. This is the work with NPOD and Professor Alberto Pugliese's team, showing that human islets from individuals with type 1 have upregulated RAGE and RAGE ligand, and in fact islet autoantibody positive individuals as well compared to controls, I believe. And of course, another Finn Dayan and a deep study in Josephine's collaboration with Professor Michael Nip. Again, lower plasma soluble RAGE is associated with a more severe disease at diagnosis. So we know, or we are still in the process of, as I said, looking into it, but some of the work that has been published in diabetes last year by our team, we know that RAGE antagonism does indeed increase functional regulatory T cells and improves pancreatic islet health, as well as arrests type 1 diabetes or autoimmune diabetes in preclinical mouse models. So we are raging against type 1 diabetes, and as I mentioned, this is a bit of a summary. So we do believe that there is a scope in preservation of beta cell function and health, and the modulation of regulatory and pathogenic T cell responses, and there is some evidence that suggesting that that is indeed the case. DC maturation and dampening of cell antigen presentation, and as well as, of course, improved glucose tolerance is some of the things that we very much believe RAGE is involved and responsible for. Reason I have trouble. Sorry. Oh, sorry. Yes. Our approach is at the moment that we are currently undertaking, and I'm not going to go into that just yet, but we are hoping to share some of our exciting data in the upcoming months. But we have some novel approaches to deliver soluble RAGE. We are repurposing oral RAGE antagonist drugs that have been tested in humans and are safe, and of course other RAGE therapies as well. Another important factor that is very, very pertinent in this situation, that RAGE gene is localized in MHC class 3 region, and it is known genetic susceptibility to type 1 diabetes. And this is a wonderful data set from the dendritic cell biologists published in Science Immunology a number of years ago, but essentially what it does show that RAGE that is indeed expressed on the immune cells, and they've used 3 different quite high quality probes. And what I suppose take-home message here, it is its expression is well above the detection threshold, and we are pretty confident that we can see in its B and T cells, dendritic cells, and as I mentioned, they mostly focus on the dendritic cell compartment, monocyte compartment. And what is interesting is plasmacytoid dendritic cells, as well as those very rare population of cross-presenting CD141 positive DCs that are so hot and so interesting, and always have been since their discovery. It is so interesting that RAGE is expressed on those cells, and this is certainly something that we are very hopeful to look into it in hopefully not so distant future, and we will again be sharing that information and that data with our type 1 community. This is a very preliminary data set, but we think is very interesting, showing the RAGE expression in PBMC of recently diagnosed individuals with type 1 diabetes. These are young people from 16 to 25-year-olds, and they have diabetes duration under 2 years. So this is a very simple flow cytometry panel. We didn't have many markers in there, but essentially what it shows is that RAGE, the CD3 positive T cells and CD3 negative non-T cells, and we didn't delineate this particular population any further. It's just CD3 negative cells, but they of course include all the myeloid cells as well. So we see that in type 1 diabetics, with recently with duration, as I mentioned, under 2 years, RAGE is upregulated on the surface of CD3 positive T cells, and this is just a histogram showing the same thing. And it's almost a reverse relationship with the, when I say myeloid, it really they really are CD3 negative non-T cells, so it's everything else. And so we think it's very exciting data, and definitely highlights the fact that this needs to be further investigated. This is a little bit of a data, a bit of a sneaky peek into what is currently being in review. And Sherman, of course, led this work, and again showing that RAGE and RAGE ligands are upregulated on type 1 diabetes, pancreatic of type 1 diabetes patients and islet autoantibody patients compared to controls. And again, hopefully this will soon be accepted and be available to our type 1 community. It's a very interesting and very important piece of work that will be shared with all of you shortly. Again, this it is he, as you probably gathered by now, is a bit of a superstar in our laboratory. He works elsewhere now, but his work, we continue to be very proud of the work done and continue this work. And this is published in Diabetes last year, that the soluble RAGE, very short term, just a 2-week therapy, delays autoimmune diabetes progression in NOD mice. And Sherman showed that that is, in fact, T regulatory T cell driven. And when you take the Tregs out of the equation, this no longer protects. So again, we are soluble RAGE. This is a fantastic piece of work. We know that, of course, sRAGE is a biologic, so we are now testing other forms of RAGE antagonist, and I mentioned the one that is orally available. We know that it is safe in humans, and I'm going to show you some of the preclinical model data. So we took that oral RAGE antagonist and we treated the mice for a longer period. It was daily for 2 weeks, and then 3 weeks thereafter, up until day 150 of life, and we monitored those mice for the disease progression. And I'm pleased to show you that it was a modest delay, but it was a statistically significant delay in disease progression in those animals. And of course, I am an immunologist, so I've got to tell you all about T cells. So and they are of course important for the disease progression, but this is again the preliminary data, and we've got many more coming. But what we were very pleased to see that in spleen of these animals, regulatory T cells were significantly increased in oral RAGE antagonist treated mice compared to control. Interestingly, in pancreatic T cell, in pancreatic lymph node, these were not changed. However, when we looked at the function of those Tregs, and this is just a snippet, we've got more data coming with the PD-1 and TIM3 expression, etc., etc., etc. But ICOS expression and TIGIT expression on those Tregs was elevated in spleen and pancreatic lymph nodes, and not quite the TIGIT, but suggestive of the fact that these Tregs are suppressive, functional, and ready to fight. From the CD8 side of things, again, an interesting observation. They were slightly up in spleens and slightly down in the pancreatic lymph node, which is very much what we want to see. And of course, when we looked at their activation status, interestingly, they show an exhausted phenotype. They're PD-1 high, and TIM3 is upregulated on those cells as well. So we are pretty confident that we are achieving some level of, and sorry, what I do need to mention, this is just after 2 weeks of treatment. So this was a separate study that we took mice after 2 weeks to have a look at the immunological profile of those cells. So it is very encouraging result, and there's a few experiments in the pipeline that we are still finishing up, and some data that we are still analyzing. But what we are very confident and hopeful that this some sort of tolerance that we are achieving here, both perhaps on the regulatory side of things as well as energy exhaustion side of things. And in terms of the improvement of beta cell function after the treatment, again, just after 2 weeks, we are seeing and insulitis is, of course, one of the measure in mice where we look at the grade 0, 1, 2, 3, and 4 based on the level of immune cell infiltration. And of course, in oral RAGE antagonist treated mice, the islets are a lot less infiltrated, which is indeed a positive result. And again, to have this just after 2 weeks is quite encouraging. Acute phase insulin release as well after oral glucose tolerance testing, it's improved in the first phase. So again, it is a very encouraging result that our treatment does do something. And just to summarize, we are very encouraged by the fact that we see the increase in functional regulatory T cells in spleen and pancreatic lymph node, and hopefully seeing some exhausted phenotype of the CD8s and reduction of insulitis and pancreata of those treated mice, as well as improvement in acute insulin release, and of course delay in these animal models. We've got, this is by no means covers the next steps. We've got tons and tons of work in the pipeline, and we're really hopeful, of course, with teplizumab, exciting result being improved in the US. We are looking into the combination therapies with anti-CD3, and of course, mouse equivalent is not quite teplizumab, but we are hoping to run some preclinical mouse model studies and have a look at different levels of T cell biology, and we are very keen to explore antigen presentation side of things, and of course dendritic cells and cross presentation and antigen presentation ability and dendritic cell maturation and all those things that are very, very important. And you know, I'm a T cell biologist, so of course I love my Tregs and I love my CD8s, but when it comes to dendritic cells, I'm very keen and to say that they can't be forgotten. So they're absolutely, it's a bit of a kind of worms with disease, and I'm sure DC biologists agree with me, but they're not the easiest cells to study, but we're going to give it a good crack. We are collaborating with Queensland Children's Hospital and Mater Young Adults Health Centre, and in fact the earlier data that I showed you from those young individuals, they are being seen in Mater Young Adults Health Centre, and we are planning to assess RAGE profile on the immune cells in at risk and newly diagnosed individuals, and we are hoping to start the recruitment in the not so distant future for that. And for that, we again planning a large study to look at lots of parameters and big comprehensive flow cytometry panels, etc. And of course, as I mentioned, testing other fancy RAGE targeting therapies for secondary prevention is in the pipeline. And again, we're very excited to be a part of this community and hopefully sharing all this data and information in not so distant future. So thank you, and I'm happy to leave some time for the discussion. And if you have any questions, please throw them my way.
Fantastic. We have the chat open, so if anyone can put something in there, some clapping in the audience for you. Excellent. Yeah, that's immediately, of course, I think everyone thinks of teplizumab and how it may be in a multi drug approach with this particular ORA. And I think that is so that has so much possibility. How are you planning to kind of run these types of trials? Are you going to do them in Australia primarily, or will there be some sort of collaboration with US or European laboratories or clinical settings?
That's an excellent question, Monica. Yes, look, we are not quite at the trials point, of course, yet. However, And as you know, teplizumab is not yet in Australia, so of course this is its caveat in itself. So in terms of RAGE antagonist trials, we have a long-standing collaboration with Professor Kevin Harold, and he is even though of course he is very busy with TrialNet and teplizumab and all those sort of things, I had a quick chat with him at IDS, and he's still very keen on RAGE side of things and work and etc. So yes, of course, collaboration is the key in any of these kind of ventures, but we will try. Yes, that's basically that. It's not really an answer, but yeah, we will do. And oh, I'm so sorry. I forgot to share with you the most important slide. Because I was too keen to hear all your questions. Yes, lots of people involved in this fantastic work, and there's not slide big enough to include everyone, but hopefully I covered everyone. But
yeah, no, it's a great collaborative space you guys have got going. I also wondered, we initiated and have been facilitating the build of a TCR repository here. It's housed on the I receptor, which is an outreach of the AIR Data Commons community, the Antibody that the Antibody Society built as a community. And so they have a very nice repository that is very harmonized, and it's standardized. And so we've asked scientists in the US so far to deposit their TCR, genetics data into this database. And Kevin Harold has expressed interest to also, add his data set pre and post teplizumab. And it seems like this situation with the ORA is really ripe as well to be a candidate, once you get those data, to perhaps add to this database. So I just want to throw that out to you, and know that it's out there, it's alive, well, evolving. People are adding 80 million sequences are in there now, and it could be really illustrative of sort of these pre and post treatment TCR profile. So it's kind of an exciting time for that. So that's out there for you guys. We're happy to help facilitate anything you need in terms of adding your data.
That's fantastic.
And then I wanted to ask, how might this treatment, we talked how it might intersect, the ORA treatment might intersect with teplizumab treatment, but is there an opportunity for it to intersect with islet implantation, of the type that Vertex is pursuing?
Absolutely. Yes. No, yes, definitely. And this is something that we are very keen. And in fact, the endport work and endport data that is going to be hopefully soon coming out, we are definitely keen to test more of RAGE in that sort of space. And it's absolutely very, 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 and in fact, it seems like everywhere you look there is RAGE. So it's basically the answer to all the questions. That's what we
in this encephalitis stage. Here's a great question. Hi, it's saying, great talk. At the time mice had pancreatic collected, how old were the mice? And was RAGE stained for in situ at protein level, or even at mRNA level by ISH? Sorry if I missed it.
So sorry. So the RAGE. Sorry. I can't see the. For some reason,
it's to everyone.
Chat, but so mice were treated from about 7 weeks old. So they're treated from day 50 up until day 150 of life, and the immunological data that I'm showing, it's just a 2-week. That's the Treg and CD8 data that I've showed. It's just a 2-week treatment, and for the rage, so we do, I'm not showing the flow cytometry data on rage, but we do have it and we are currently analyzing. And it seems like there is a upregulation of rage. And it seems as though you need a little bit of inflammation at the start for it to be able to work later on. In fact, some of the cytokine data seems to suggest that as well. But if the question relates, sorry, I can't see the chat for some reason, but if the question relates to the human eyelid, I don't think it relates to human eyelids. It relates to mice.
Yeah, the
right?
mice. How old were the mice, and was rage staying?
So they were, yeah, so they were 9-week-old when the immunological studies were run, and the encephalitis was also done at that point.
Great. Yeah, I just want to know whether you could see like staining for mRNA or protein. Very speculative, this question, very speculative, but how do you think this might translate in terms of delaying type 1 diabetes in humans? How do you expect the NOD mice findings translate to humans? That's another question.
Yeah. So again, for those of us who with just the IDS, there was a lot of talk about staging of the disease, and of course, this is a very hot discussion at the moment, is when to treat. And I think Lucien Chateauneau and certainly Chantal talked a lot about that. Is less is more? Do we need to hit earlier? Do we need to wait until this inflammation and autoimmunity sets in? And it is a very difficult decision to make, and in terms of even running this as a clinical trial, as to when do we intervene. But our preclinical mouse data suggests that we do need to start in that pre-diabetes stage, and of course this would be a preventative therapy. So this would potentially be. However, we do know that the targeting rage also does reduce inflammation. So perhaps there is a scope to look at the later time points. And these are all great questions, and there is also more questions than answers for sure.
Yeah, well, it sounds like it'll all be about, determining the windows of treatment.
Exactly.
And whether or not there are different in different endotypes. But it's exciting, and it's still, the data you've shown are quite exciting, and the data we shared or we saw from Sherman Long and Josephine Forbes a few weeks back was also quite interesting. So we're really cheering you on from here and hoping that that everything accelerates according to plan. And we'll look forward to some of these new papers coming out soon too. We'll be sure to highlight them and amplify them, to our audience across the globe, because I think it's really an interesting approach, and it intersects with many different other approaches sort of happening right now. So it's very timely. Okay, well, thank you so much. I really appreciate you coming and talking, and we will we'll be in touch again. Thanks again. Have a great rest of your day.
Thank you, Monica. Thank you.
Australia. Bye-bye.
Thank you, everyone. Bye-bye.